DNA, the sort of actual information that is contained within the DNA. And we understand that DNA is a molecule in the body which carries genetic information that we inherit from our parents. Now, a mutation is a change in the DNA which can either be inherited, that we get either from our mother or our father, or it can occur after birth through environmental exposure to things like tobacco smoke or foods, or just because of aging. And some of these mutations can result in an incorrect amount or type of protein made by the DNA, by the genes. And these kind of changes can actually result in disease and in some cases cancer. So with that as a background, what we really understand now about cancers is that they are in fact all genetic, which is to say there's something that goes wrong with the genes that makes a cancer. And there are sort of two different types of pathways that we understand. We understand now that cancer is really a multi-step process that occurs in the lifetime of a cell. And in some cases, the cells, as I say in the first arrow, an individual can inherit a mutated gene, a gene which already has in the sequence of the sort of alphabet soup, there's a switch or a change or a kind of a mix-up in the alphabet soup, and that results in a gene that makes a protein that, as I had explained, is sort of doesn't work properly, or there's an incorrect amount. And that person already starts off their life kind of with one foot down along the pathway towards cancer, because that particular gene is in every single cell of their body. And that individual can, over the course of their lifetime, accumulate other mutations that occur in their cells, which are called somatic. Somatic meaning it only occurs in particular cells over the course of time. But those accumulated events between the mutation they inherited and the mutations that occur just in the process of them living their lives can result in cancer. Alternatively, and much more commonly, an individual just accumulates a certain series of mutations over the course of their life, and that can result in cancer. But both of these pathways we understand are very important in how a cancer can result from genetic mutations. Now, in some cases we understand, and I put this picture up just to sort of show a sort of image, we know that in some cases cancer can run in the family, and we've learned an awful lot about some of the common mutations that occur in these individuals, and I'll speak about that a little bit later with respect to some of the research that we know and also some of the new research that's already come out of the TCGA project. But to explain a little bit more in global terms, what the Cancer Genome Atlas project basically is designed to do is to map all of the mutations that occur in more than 20 different types of cancer. And it's using a whole range of amazing new technologies and amazing cooperation between numerous different institutions and scientists. What the pilot project began in about 2006, and that project is designed to focus on lung and brain and ovarian cancer, which collectively account for about 210,000 cancers per year in the United States. And the whole goal is to create literally an atlas to help us understand what actually goes wrong in the cancer cell. What is it that actually turns a normal cell into a cancer cell? This is a collaborative project involving more than 12 institutions, including governmental agencies like the National Cancer Institute, the National Institute of Health, as well as industry, as well as academic centers. And the goal is ultimately to create this atlas of key genetic targets or genetic signatures in cancers that will help us hopefully define and find new prevention, diagnostic, and treatments for cancers. So some of what I want to explain is how the TCGA project differs a little bit in its approach from what we've always relied upon in taking care of patients with cancer. And this is a slide that I've borrowed, which kind of shows that once upon a time, in the old way, we took a sample of the patient's cancer tissue, we looked at it underneath the microscope, and we tried to sort of define the patient's disease by the features that we could see underneath the microscope. And this is information that may be familiar to many of you, in which we describe the pattern of the cancer, how or where the cancer has spread to, which is called the stage. And we relied upon that very basic information to predict the behavior of the cancer and to determine what would be the appropriate therapy. But the new way, which is really what we're going to see coming out in much more detail as the Genome Atlas project continues, is using a whole constellation of ways of actually understanding the proteins and the genes, which genes are turned on, which genes are turned off. So a whole different level of exploration in the cancer, which will then give us better insight into what was actually the responsible mutation or series of mutations that made that particular cancer. In other words, what were each of those accumulated little ticks on that pathway that ultimately resulted in the cancer? Because as we better understand the specific errors in that cancer, we can define and describe and choose much better therapies that will presumably work better for the patient. So I would say in summary, what the TCGA is all about and sort of the new approach to cancer genetics is looking a little bit more under the hood of the actual cancer to understand better what are the particular errors or things that are wrong, so that we can have a better sense of how to use better treatments, either predicting which ones will be more effective or avoiding those that we think will be less effective. And this is really very exciting because this science has really not worked so well until this big collaborative effort to get patients who are willing to donate tissue and multiple laboratories that really are very good at these kind of analyses and collaborating amongst all of these different centers. That's really where we hope to see great progress. So I share with you here a slide. This is a paper that came out in Nature in looking at ovarian cancer and some very, very preliminary results that looked at about 500 women who had the most common type of ovarian cancer, in which there was a comprehensive genetic analysis, including information about how each of those patients did, what chemotherapy they received, how did they fare with the chemotherapy, did their cancer come back. And this was really important because it gave us the ability to sort of link what was the particular mutation that the cancer had with the actual behavior of the cancer. And lo and behold, what we found is that there were some very common mutations in many of these cancers. The P53 gene was mutated in almost all of the samples, and the BRCA1 and 2 gene was mutated either as a somatic in the actual ovarian tissue or a germline mutation that individuals had inherited from either their mother or their father in about a quarter of patients. But there were some other genes that emerged that also were common, occurring in about 2 to 6% of patients. Some of these genes we've already studied and have a little bit of insight into. But further, there were additional additional information about some of the pathways that were incorrect or didn't work properly in ovarian cancer, including both signaling pathways and homologous recombination pathways, the significance of which is just it gave us a little bit of an insight into some of the things that are wrong, some of the errors, which may give us some targets in the future for effective treatments for women with ovarian cancer. I think it will be not a surprise to anyone when I show you the next picture that there was a little bit of disappointment at the wide range of things that are wrong in ovarian cancer. And what is clear is there's no one dominant target or no one dominant mutation that will be the sort of band-aid for everyone. And that's sort of depicted on this next slide, which I think in some ways looks like a little bit of complicated wallpaper. But it does begin to give us some sense through the very detailed genomic analysis of ovarian cancer samples, what kind of mutations are seen, how often, and how we might exploit this information to devise better therapies for women with ovarian cancer. So moving on then to a little bit of specific information and some examples of how we've used some of the information that we understand about genes in ovarian cancer that are important and moving towards personalized medicine. We do know that one of the very common causes of that hereditary or sort of family cancer, ovarian cancer and breast cancer syndrome are the BRCA1 and BRCA2 genes. Now these are genes, or rather looking at the entire sort of pie of women with ovarian cancer, which is depicted on the left hand of the screen, we see that most patients in blue have ovarian cancer just because of bad luck. They're the people who just for some reason, there was an accumulation of mutations over the course of their lifetime that resulted in ovarian cancer. But about 15% of women seem to actually develop ovarian cancer because they inherited a genetic mutation in the BRCA1 or 2 gene from either their mother or father, and we understand that these genes are very important in keeping the cell's DNA replicating or dividing properly. And if those genes are mutated, there can be, as I had alluded to earlier, sort of already one step along that pathway towards developing a cancer. And we understand that of all of the patients who develop hereditary cancer, the BRCA1 and 2 gene are in fact the dominant causes of these sort of cancer family syndromes. Now, in general, we know that BRCA mutations occur in about 1 in 400 to 800 people, although there are some groups of people that have this BRCA mutation more commonly. And in the United States, one of the most frequent groups we see is in Ashkenazi Jews, in which the BRCA mutation is seen in about 1 in 40 otherwise healthy and unaffected individuals. And what we've understood now over time is that the BRCA, the individual who has an ovarian cancer, who has a BRCA mutation, has certain kinds of DNA damage that occur just over the course of every single day that their cell is not very good at fixing because it doesn't have that sort of police gene, the BRCA gene, to help keep the cell in line and repair the inevitable errors or damages or sort of insults that occur to the DNA. And it turns out that there is a drug called a PARP inhibitor, which we've now discovered is another way of sort of stopping or shutting down the cell's ability to repair DNA. And it so happens that in individuals who have BRCA mutations, lo and behold, when they take this PARP inhibitor, it seems to be even more effective in them than individuals that do not have the BRCA mutation. Simply put, the BRCA mutation carrier, it has a better response to the PARP inhibitor because of the precise genetic abnormalities or mutations that occur in their cancers. And this is a sort of good example of where we're trying to ultimately move with personalized medicine. Can we figure out, are there certain drugs that work better in certain individuals because of their genetic mutations? And again, while this this picture rather is a little complicated, it's trying to basically illustrate one example of personalized medicine that has already shown great promise in ovarian cancer by exploiting the cumulative loss of the cell's repair tools and basically this concept of the BRCA mutation carrier being more susceptible or having a better response to a particular type of treatment is exactly where we hope to go as we learn more information through the Cancer Genome Atlas project. This picture, I think, is a good sort of reminder that as we better understand the specific mutations or errors, we can devise personalized therapies or targeted therapies, which ultimately are designed to treat the cancer cells but leave the rest of the cells in the body alone. In other words, try with a targeted therapy to find a drug that, like a magic bullet, aims right for the cancer cell but leaves the other cells in the body alone, so that the patient ultimately can have fewer side effects and more effective treatment, which is really where we want to move in the era of personalized medicine. So some very simple picture examples of some of the tools that we are beginning to see in cancer genetics and genomics. One concept, which is depicted here, is that we understand, and this harkens back to my analogy of the old way in which we would identify a patient's cancer and stage and grade and try to predict how would that cancer behave and what might be the best treatments for that cancer. And you can see the patient group is depicted in the center here as individuals that are many different colors. And you could argue that the old way that we've relied upon recommending certain treatments to a patient is an incredibly simplistic picture, because the truth is there are some people for whom the drug will not work and it will actually give them a lot of side effects. That would be the individual who has no benefit whatsoever and they have toxicity or a lot of excess side effects. That would be the people that are listed in white. There are other people for whom the drug may be effective, but it actually causes too many side effects. There are other people for whom the drug is going to be very effective and have very minimal side effects. So as we understand, we need a better way to figure out who's in the red group and the white group and the yellow group and the green group. And this is really where, as I say, more insights into the actual genes that cause the cancers will allow us to figure out who's in which group and ideally pick the very best treatment, or at least avoid treatments which are going to be ineffective and cause a lot of side effects. So some examples of this now, this concept of targeted therapies and personalized medicine that we're currently using already include breast cancer. And one example in particular is the HER2/neu gene. This is known to drive certain cancers. And what was discovered is that an antibody, an actual treatment that can block the gene and thereby decrease the extra copies of this particular gene product, which seem to make the cell grow and grow, is actually a very effective therapy in breast cancer. And this is held up as the real sort of shining example of where we can go with personalized medicine. Again, if we understand what's the specific defect and we understand the specific treatment, and that's sort of depicted with the yellow and green picture in the right upper corner, if we can block that particular specific gene that's causing the cancer, why then we have a very effective therapy which will minimize side effects to all of the other cells. We also see now in breast cancer that it's fairly common to use a battery of molecular tests which have been shown to very nicely predict whom is at risk to have their cancer come back. So we're now trying to use more of these sort of assays to help guide therapy when a physician talks to their patient who's just been diagnosed with breast cancer and has had the breast cancer treated or has a piece of tissue. Should this patient get treatment, or does she in fact not need treatment because her molecular tests, this battery of molecular tests, would actually predict that she does not have a very high risk at all of her cancer coming back. Other examples that we see in lung and colon cancer, again, are distinct gene expression patterns or enzyme deficiencies can actually predict who's going to respond to certain treatments and who isn't. And again, using this information, we can avoid treating a patient with a drug that might cause a lot of toxicity and not very much benefit, and rather direct our treatment specifically to those patients who seem as if, based upon their gene expression patterns, they should have a good response and not too much toxicity. And I think the sort of magic example that all of us hold up is what we saw with leukemia, and a form of leukemia called CML, in which there's a specific enzyme, excess enzyme, that actually causes the leukemia. And there was a specific drug called Gleevec, which was devised that blocks that excess enzyme. And we have seen in this example that this one drug has really been the magic bullet for many patients with this particular enzymatic deficiency in CML. And I would just pause here and again reflect upon the fact that the information that we have so far from that publication in the Genome Atlas Project in ovarian cancer suggests that ovarian cancer will not be as simple a ball of yarn to unravel as a disease like leukemia, in which there's just one dominant target. It's pretty clear that ovarian cancer is much more complicated, and we will need to think outside of the box and perhaps even use multiple different targets, again, depending upon what are the specific mutations in a patient's ovarian cancer. So I would like to conclude with this slide, which just shows, again, in a schematic, an awful lot of list of very difficult to pronounce compounds, but based upon available research, these are some of the compounds that are being tested right now, and we can understand from, again, where so the information that we've learned about cancer genes and what makes a cancer cell different from a normal cell, we understand that there are certain genes that are either too much activity or too little activity, or pathways in which there's something that's gone wrong with the pathway. And many of these drugs, as depicted here by whether they're antibodies or many different mechanisms of action, how can they specifically fix the mutation in the ovarian cancer cell, hopefully minimizing toxicity to the other cells and ultimately resulting in some activity or put another way, some benefit to the patient by controlling her ovarian cancer. So the last thing I'd like to share with you briefly, because I think it's incredibly valuable, is some information that is on a website from the Cancer Genome Atlas Project. There's a very nice brochure, and I have borrowed this for you because I think it's a very good reference and guide and very nice introduction to the concept of the Genome Atlas Project, what they hope to achieve, and some much more nice detail about the project, what it involves, and their anticipated timeline. So I will stop at this point, and I'm happy to entertain any questions. Again, I apologize for the somewhat accelerated nature of to share right now, and much cause for enthusiasm insofar as what we will learn and where we will go with treating ovarian cancer in the future.
It's wonderful. Thank you, Dr. Katz, very much for your very interesting presentation. It was great. It was a lot of great information, and we do have some questions that have come in. You talked about PARP inhibitors, and we've gotten some questions related to PARP inhibitors. So I'm wondering if you can comment on the data that's out there in terms of how effective are PARP inhibitors for BRCA, for patients with BRCA mutations, and also related question: what's the current state of research regarding the use of PARP inhibitors for ovarian cancer patients that don't have BRCA mutations?
That's an excellent question. So let me begin with the PARP inhibitor data. And there was, first of all, the initial data in PARP of PARP inhibitors focused on patients with BRCA mutations, but there was just a publication in the New England Journal of Medicine, which is a very important journal for us in medicine and certainly in oncology, looking at using the PARP inhibitors in individuals who had serous or high-grade cancers, irrespective of their BRCA status. So we do have some very good data that shows that this is an active drug. This particular drug was called olaparib, and this was a drug that was used in patients who had ovarian cancer that had recurred and been treated with a chemotherapy regimen, including platinum agents, which is sort of our go-to sort of that's our kind of really good working horse drug in ovarian cancer. And in the entire group of patients with recurrent ovarian cancer who'd been treated with a platinum agent, it did look very promising that the PARP inhibitor was able to keep their cancer away for a longer period of time than those women who got placebo. So that's a very encouraging result and mirrors to some extent what we've seen in BRCA mutation carriers, that really there does seem to be some very good activity, or simply put, the cancer seems to respond to these drugs. Now, things are somewhat complicated by the fact that the company that was making that particular compound, at the present time has decided they are going to sort of take a little time out, and they're not really expanding any studies at the present time using this particular compound in patients with ovarian cancer, whether they have BRCA mutations or they don't. Now, I should add there are other compounds like the olaparib, which was studied in this particular study. There are other similar compounds that are being studied in clinical trials right now, but for the present time, I think we're just going to have to wait and make sure that these other PARP inhibitor type drugs are just as effective, and undoubtedly to begin with, we're going to see more of those studies in BRCA mutation carriers, but I expect if there continue to be promising results, they will trickle out and to the larger population of women with high-grade serous ovarian cancers.
Great. Okay, thank you. Now, you mentioned that there's some predictive possibilities for treatments, for personalized medicines for certain kinds of cancer, like lung cancer. Is there a similar blueprint for ovarian cancer? That's
an excellent question, and not really, not yet. Again, that's really what's going to be very exciting about the Cancer Genome Atlas project. Will we start to evolve, or will we start to develop, again, a sort of genetic signature that defines for us, these are the people who tend to not respond, whose cancer tends to come back, and these are the people who seem to respond very well? We don't really have a clear sense of that right now. One of the genes that we know has been shown to be important in sort of determining the outcome or how a patient will do are those BRCA genes. And again, data based upon the Genome Atlas project that linked up tumor samples with the clinical information about how patients did with their treatment found that, in fact, BRCA mutation carriers, most notably BRCA2 mutation carriers, tended to have the very best response to chemotherapy and tended to live longer than women who have BRCA1 mutations and women who do not have BRCA mutations. So again, this is a very important study that begins to, again, sort of shed light or, as I say, kind of open up the hood and let us actually really look into the machinery and the motor of the ovarian cancer cell and understand, okay, what is it that makes this cancer cell tick? How can we disable the cancer cell? And some of this data is very promising to give us more information about is there a genetic signature that predicts outcome and potentially response to chemotherapy.
Okay, great. Thank you. There's another question about PARP inhibitors are coming in. In terms of the availability of PARP inhibitors, can you talk a little about if people are interested in finding out more how they might access it? Is it
Sure.
exclusively through clinical trials at this point?
It's exclusively through clinical trials at this point, and a website that I would, or a number of different ways in which to obtain information, but the National Cancer Institute has a website that gives information about clinical trials, and I would encourage anyone listening who's interested to look on, literally, you can look online and get information about, on nci.gov, the clinical trials that are available that include PARP inhibitors. There are select institutions really across the country that are participating in these trials, but at the present time you can only get a PARP inhibitor if you're on a clinical trial.
Okay, great. And I just want to interject a little bit about OCRF. We have a great clinical trials matching service available on the OCRF website where you can go in, put in information about your treatment history, and it'll help you link up with a trial that you qualify for. So you can find that on ocrf.org. Okay, we have more questions. One question is about the use of targeted therapies for, or rather, the use of the Cancer Genome Atlas and this kind of information for the prevention of cancer. We've been talking a lot about it in terms of treatment of cancer, but are there any possibilities for the prevention of cancer, ovarian cancer?
Absolutely, absolutely, because again, if we understand not only the kind of panoply of genetic mutations that occur to make a cancer cell, but if we can even unravel things and understand what was the first thing that went wrong, what was some of the early mutations that occur, then we can absolutely design trials aimed at prevention. If we can prevent that mutation from occurring to begin with, if we understand that a particular pathway of genes that may be responsible for how one cell moves to another place, how a particular cell draws its nutrients from the bloodstream, then we can actually talk about targeted therapies, but prevention, and even better diagnostics, because I think an incredibly important point that is very familiar to this audience, what continues to be so difficult for us in ovarian cancer is that the majority of women are diagnosed with their cancer when it has already spread outside of the ovary. In other words, they have advanced stage disease. And we know from the data that if we can find more women with ovarian cancer that is early or is still confined to the ovary or the pelvis, we can actually treat their cancers quite well and have very similar success rates to patients with breast cancer, where we have a lot more familiarity with the success of treatment for early stage disease. So part of what the TCGA will be aimed at is identifying improved diagnostics for ovarian cancer, which ultimately really should make a huge impact on how patients survive with ovarian cancer.
For people who are interested in contributing to these sorts of studies, how do they go about, say, donating tissue? Is that something, is conversations they initiate with their doctor? That's a question that came in.
Yes. Now, and remember that they have very rigorous criteria for getting tissue from patients. So specifically the institutions that, at least in the pilot project, have been carefully selected. And this is, again, all available on the Cancer Genome Atlas website, which I have up for you on the screen. You can see which of the institutions that are participating. But basically, at the present time, it's very important that the tissue be processed and sort of sort of processed, and that the quality and quantity and the way that the tissue is handled be very rigorous and very careful, because of course we want to have accurate information. So at the present time, there are just a select number of institutions that have the machinery and the infrastructure to process these samples. But these websites you can find on the web. Rather, you can find these institutions through the websites that I've provided
Great.
for you.
Perfect, thank you. Another question, and then we'll be wrapping up soon, which I'll just read verbatim because it's clear. Do you envisage that in the future that targeted therapies will have significant activity as a monotherapy and not require being combined with chemo? And is there a risk that we're neglecting chemo/cytotoxic drug development in the rush to targeted therapy?
Excellent question, and I think, I would submit to you, and in a somewhat jaded response, we've been using the same sort of ingredients to make this sort of cake for a very, very long time. We've made some incremental progress, to be sure, but I think that it's very much time to think about thinking outside of the box. We need some new ingredients to put into the mix to make a better loaf of bread. And so the combination, and remember, the TCGA will simultaneously shed light upon information with cytotoxic treatments, because we do have some fairly good understanding about how chemotherapy works and which of the genes and pathways the cytotoxic chemotherapy seems to affect. And in some cases, we may see that the best outcome will be combining a cytotoxic with a targeted agent. And that's where many of our trials right now in some of the cancers, breast and lung and colon, that have a little bit better sense of some of the gene derangements or alterations. That's very much the philosophy that's been used in some of those cancers, and I fully expect that's what we will see in ovarian cancer. But I think the TCGA, as I say, is basically going to shed light on information that will be helpful not only for targeted therapies, but for cytotoxic therapies and for the combination, so that we can really push the envelope a little bit further along with respect to better outcome for our patients.
Gotcha, okay. One more question someone submitted. He says, I'm aware of labs in the United States that upon being provided with a large enough tumor sample can expose tumor cells to 30 cytotoxic drugs to see which are the most effective. And he's wondering what you think of this strategy.
So that is a good question, and we do have some there are some companies at the present time that provide these sort of chemo resistance assays. And while it sounds a very appealing concept, unfortunately the data has not really shown that the correlation between what is predicted by growing cancer in little petri dishes and treating it with chemotherapy, the correlation with how the cancer behaves in the petri dish in the laboratory and how the cancer behaves in a person's body is not very good. In other words, the drugs that would be predicted to work quite well in the petri dish do not always work in the human. Part of that undoubtedly is that the petri dish is just too simple of a model in which to study the behavior of the cancer. And again, the TCGA project is aimed at really dissecting out and understanding at many different levels, not the genes, but the genes that are turned on and off, and the proteins, and the complex interaction between all of the genetic processes to understand what's actually happening in the human body. So while these assays are commercially available, they have not really been very helpful in predicting who responds best to which treatment. At least at the present time, the science is not yet there, and currently our societies do not really endorse using this testing as a method by which to choose treatments for patients. But I think it is the beginning of a very good concept, which we hope to see expand and ultimately give us better results in the future.
Great. Okay, last question. I wonder if you can comment on some possible implications of this work for patients with multiple cancers, say breast and ovarian, and those with recurrent cancer.
Well, again, I think For drug resistance, right. So, unfortunately, the nature of the beast with ovarian cancer is that the majority of time it tends to recur, and the majority of time, ultimately, it is the resistance of the cancer. The cancer just becomes basically resistant to any of the chemotherapy drugs, even drugs that had previously worked. And that's the part that's really vexing, because once upon a time the treatment that seemed to work quite well no longer does. And this is where, again, the long-term follow-up in projects like the TCGA, which will look at patient samples not only ideally when they were first diagnosed, but even when their cancer came back, will provide us with a really unique opportunity to see what changed even in the genetic makeup of that cancer between the recurrence and when they were first diagnosed, and can that give us some further insights into which drugs may be helpful and which drugs are not, which pathways are activated, which pathways are not, ultimately, hopefully, with the intent of devising better treatments with less toxicity that are targeted to the specific mutations in that particular cancer, because we know that cancer is, after all, a living cell, and it's capable of changing itself and responding to its environment to try to basically survive. And so we need to understand a little bit better what happens over the course of the cancer's life cycle, even when it comes back and manages to sort of become resistant to chemotherapy. And all of this, we remain very optimistic, but the TCGA project, we do think will shed some light on ovarian cancer resistance.
Okay, thank you. I want to thank you, Dr. Cass, for your really excellent presentation. And I want to thank everybody for calling in today. I just want to make note of a few things. This presentation will be available on the OCRF website after the webinar. And also, everybody please mark your calendars for May 23rd. That's the date of our next webinar, where we'll all be hearing from Dr. Andrew Burchuk at Duke University, and he's going to speak about screening and early detection of ovarian cancer. So stay tuned for that and for more information about other upcoming presentations. In the meantime, please take a minute to fill out the survey that will appear on your screen at the conclusion of the event. Your feedback is really helpful to us as we go forward and plan future webinars. And for more information about OCRF, you can always visit our website at ocrf.org. So thank you again to Dr. Cass for her time. And thank you to everybody for calling in, and have a great evening.